Hello everyone, welcome you all in this video lecture. So in this video tutorial we are going to discuss about CRISPR-Cas9, which is a very important method in the genome editing. So let's start with the introductive part. So basically the CRISPR-Cas9 is considered as a powerful genetic tool which is used in the process of genome editing. So let's understand first that what is the genome editing. So the genome editing is also called as gene editing or gene engineering. And the genome editing is the way of making specific changes to the DNA of particular cellular organism. So we know that what is the DNA. So DNA acts as a genetic material in most of the cellular organism, and DNA have genes in its structure. So we know that what are the genes? They are the nucleotide sequences and they are considered as unit of hereditary information in all cellular organisms. So these genes are the responsible factors for protein synthesis, translation and all the other cellular activities. And these genes are also responsible for inheriting the traits from parents to offspring. So there are several diseases or genetic or inherited disorders which caused due to malfunctioning of genes or due to some disorders from genes, and they are inherited from parents to their offspring. So with the help of genome editing or several editing processes in these genes, these malfunctioned genes can be replaced or the proper genes can be inserted in the place of these malfunctioned genes so that these diseases can be cured. So basically, the genome editing is a type of genetic engineering in which the DNA is edited with the help of several functions which are like insertion, deletion or modification in the genome of cellular organism. Now the CRISPR is an acronym for clustered regularly interspaced short palindromic repeats. So basically the CRISPR sequences are DNA palindromic repeated sequences which are found in DNA sequences, and these DNA sequences are commonly found in the prokaryotic organisms like bacteria and archaeobacteria. And in these organisms they help them for the preventing them, and they are derived from DNA fragments of bacteriophages that have previously infected the prokaryote. So because we know that prokaryotes are infected by bacteriophages, which are also a type of viruses which engulf bacteria. So from these bacteriophages, these DNA sequences are isolated or derived and they are used as a genetic tool. Now let's understand some characteristic features about the CRISPR-Cas9. So we know that CRISPR are some palindromic repeats and they are found in approximately 50% of sequenced bacterial genomes. Now let's understand that what is the Cas9. So here the Cas9 is associated or termed for CRISPR associated protein 9. So here the Cas9 or CRISPR associated protein 9 is a type of protein which acts as an enzyme. So basically it is an enzyme which uses CRISPR sequences as a guide to recognize and cleave specific strands of DNA that are complementary to the CRISPR sequence. So this editing process has a variety of applications including biological research, treatment of several genetic diseases and many other applications. So the CRISPR sequences together with Cas9 proteins used as a technology, or they are used as a genetic tool due to their tendency to cut and recognize the specific strands of DNA, and this tool is called as CRISPR-Cas9. Now let's discuss about the discovery of this technology. So the CRISPR-Cas9 technology was developed by Emmanuel Carpenter and Jennifer Doudna, and both the scientists were awarded by Nobel Prize for their discovery in 2020. Now the question arises that how the CRISPR sequences can be identified in the bacterial genome. So basically they are widely distributed among bacteria and archaeobacteria, as we have seen in the data in our previous slide that they are found in 50% of the bacterial genome. So as we have discussed that CRISPRs are palindromic repeated sequences. So their most common and notable feature is they have repeating spacers and direct repeats, and that's why they can easily identified by several methods. So thus they are easily identifiable sequences. So now let's understand about the working mechanism of CRISPR-Cas9 or how this technology works. So basically the CRISPR-Cas9 uses a combination of an enzyme, which is Cas9 protein, and a guide RNA. And here the CRISPR sequences work as a guide RNA because they have the specific recognition sites. So the enzyme, which is called as Cas9, act as a pair of molecular scissors. So we know that we have a class of enzyme called as restriction enzymes or restriction endonucleases, which are also considered as molecular scissors, and these enzymes have the ability to cut the strands of DNA at a specific location in the particular genome. So that bits of DNA and then can be added or removed. Now, the guide RNA is the another component of the CRISPR-Cas9 technology, and it is a piece of RNA which is called as gRNA or guide RNA, and it consists of a small piece of pre-designed RNA sequences which can be approximately 20 bases long and located within a longer RNA scaffold. So this guide RNA is a part which binds to DNA, and the pre-designed sequence of this guide RNA Cas9 to the right part of the gene, and this makes sure that the Cas9 enzyme cut at the right point of the genome. So here in this technology the guide RNA guides the enzyme to make cut in a specific sequences. So that's why the guide RNA is named as guide RNA, because it recognizes the specific sites in the genome, and in this specific site the enzyme used as a molecular scissor and it cut in the specific nucleotide sequences in the DNA, and after that the genome can be edited or inserted a required gene instead of this gene. So now let's understand the whole mechanism of the working of CRISPR-Cas9 with the help of this diagram. So here we can see this is the double helical structure of DNA molecule, and this is the guide RNA molecule. So here the guide RNA molecule recognize the specific site or recognize the specific nucleotide sequences within the DNA molecule, and after that the enzyme or the restriction endonucleases come and make cut in both the strands of DNA in the specific nucleotide sequence. And here we can see the double strands of the DNA breaks with the help of these restriction endonucleases. So at this stage the cell recognizes that the DNA is damaged, so it starts its repair mechanism. So basically the cell have two repair mechanisms which are named as non-homologous end joining, and another one is homologous direct repair. So in the condition of non-homologous end joining, it didn't need any template and it joins or repair the DNA. But in the case of homologous direct repairing, it need an external DNA as a template, and with the help of this template this mechanism repairs the DNA, and thus the CRISPR-Cas9 mechanism works in the process of genome editing. Now let's understand that how the technology of CRISPR-Cas9 is developed. So basically, we know that CRISPR are the repeated sequences found in prokaryotic organisms, especially in bacteria and archaeobacteria. So several bacteria have a similar gene editing system as CRISPR-Cas9. And these bacteria use these sequences to respond to invading pathogens like viruses and bacteriophages. So these bacteria are used to protect themselves from these pathogens. And scientists adopted this system so that it could be used in other cells like from higher animals, higher plants, including mice and humans. And they use this system to improve the genetic variations and to cure some genetic disorders which cannot be cured by conventional methods of medicine, and several other applications which are beneficiary for the humans. So now let's understand that what are the applications and what is the significance of CRISPR-Cas9 for humans. So basically, the CRISPR-Cas9 is the genetic tool which has a lot of potential as a tool, and it is used to treating a range of medical conditions or a range of genetic disorders which can be inheritable or which are inheritable from parents to their offspring, and several other genetic disorders which are caused due to several types of point mutations in the genome of particular cellular organisms. The technology is also used to treat cancer, hepatitis B, diabetes, thalassemia, and many other genetic diseases. Now, the CRISPR-Cas9 also used or involves in the process of genome editing. Now we know that what is the genome editing, and the process of genome editing use several types of genetic tools for the process, and it is also a type of genetic tool which involves in editing of particular genome of a cellular organism. And the CRISPR-Cas9 technology is also used as a diagnostic tool for several diseases and several disorders. So these are the applications and significances of CRISPR-Cas9 and also genome editing. Besides this, there are several limitations also for the process of gene editing or genome engineering. The concern is the ethical concerns with these processes, and there are many other concerns or disadvantages for this process. So in this video we have discussed about the CRISPR-Cas9, its mechanism and its significance for the. Thank you.